Andrew Cox is going to present our next talk. He's actually, out of like a big pool of submissions, our selected talk for a submitted talk. And so he's going to talk to us. So he came down from Dallas, from UT Southwestern. And so he will be talking to us about accurate automated classification of prognostic genetic abnormalities in AML and APL. So, yeah.
Yes, thank you. I know that's a bit of a mouthful. So thanks for saying it so I don't have to. I'm honored to be down here and speak with a panel of illustrious speakers, so thank you. And yes, I'm a graduate student at UT Southwestern Medical Center, and I'll present today about some work I've done for automated detection of cytogenetic abnormalities related to AML and APML. A little bit of background about the biology of how this disease develops. So the myeloid and lymphoid lineages develop from the hematopoietic stem cells, and we're going to be solely focusing on the myeloid lineage today, where AML pertains to. And APML is a specific subtype of AML that is developing in the promyeloblasts prior to becoming neutrophils. So acute myeloid leukemia, its prevalence is about 21 in 100,000 people, and it has many varied prognosis according to genetic subtypes. So some have favorable prognosis, whereas others don't. APML, acute promyelocytic leukemia, again, is a subtype of AML. It's about 5 to 10% of AML cases, depending on the region. Some places, like Argentina, it's actually cited as up to 50% of AML cases, so that does vary. But it went from being a deadly disease that resulted in very premature death to one that can be treated with quite high complete survival rates or complete remission rates and 5-year survival if detected and treated early. And so the hallmarks of APML is this PML-RARA fusion, which occurs from this translocation between chromosomes 15 and 17, whereby you get this PML-RARA gene fusion, and that causes a differentiation block in the promyelocytic lineage, and that accumulates. And so the standard of therapy is a dual treatment with arsenic tetroxide and all-trans retinoic acid, and that actually will cause the degradation of this oncoprotein and result in these favorable outcomes. And so some other genetic abnormalities that I'll be mentioning today include translocation between chromosomes 8 and 21, the RUNX1 and RUNX1-T1 rearrangement. This is generally a favorable prognosis in AML, and it's correlated with several subtypes. There's over 22 emphasized of these subtypes according to the World Health Organization, so I'm just highlighting a couple. Another one is the inversion of chromosome 16 and the translocation of self-location of chromosome 16. And this is associated with the core binding factor protein and the myosin heavy chain 11. And this also has a favorable prognosis and is associated with certain phenotypes of AML. And so the point I'd like you to take away from this is that the presence of chromosome abnormalities is one of the most important criteria for leukemia diagnosis and management. And but the diagnosis of these diseases is a multimodal approach, of which cytogenetics is just one component. And so we'll just be focusing on that today and not the clinical variants and molecular sequencing. And so this is a general process flow of how cytogenetics will take place. So you get a sample of marrow or blood, culture the cells and arrest them in metaphase, and then you can actually fix them and spread them on a cell and then band them for imaging. And they'll be, in many places, automatically imaged and then analyzed by a cytogeneticist. And so this talk is mostly just focusing on this karyotyping portion and how the analysis can be automated there. And so why machine learning for cytogenetics? Well, hundreds of these cells are being imaged in the cytogenetic core and they go unexamined. So typically, a technician will find 20 cells that are high quality that the cytogeneticist can analyze, and only 2 of those cells will actually be analyzed. So it's very hard to find like rare subclones by cytogenetics alone. And typically, this is going to take anywhere from 5 to 30 minutes to analyze per case based on the complexity of the cytogenetics. And so leveraging machine learning to automatically detect these abnormalities could help improve efficiency there, reducing turnaround time. Can also reduce variability, serving as a clinical decision support system, and help analyze more data that's already being collected. And in the context of APML, we're talking about a disease that days can make a difference in somebody's life. So being able to detect these abnormalities quickly can actually result in quicker treatment and reducing the likelihood of hemorrhage. So a little bit of background on convolutional neural networks. So they apply a filter to some sort of input, and they're one way of very popular architecture for deep neural networks. So on the right-hand side, you see the input of a hand-drawn digit 2 that goes through several layers of many convolutional filters, and then actually gets spread down to a fully connected neural network. And there's some sort of output layer that's outputting what the label it thinks it is for the input. And so this talk will just deal with CNNs today. So for any computer vision or deep learning historians that care about that, cytogenetics is actually one of the first known instances of a computer vision task. So back in the 1960s, there's an algorithm developed to actually identify chromosomes based on their relative length. And so you can see that it actually does pretty good. The top row is the cytologist annotations, and the bottom row is the algorithm. And so overall, it does pretty well, just looking at the relative length in this case. But it was a long time until CNNs came along. But once they did, they took this task and ran with it. And so there are many models that have been trained to classify normal chromosomes, and these are in a review in 2020 demonstrating really robust performance across data sets of tens of thousands of cells. And so we have many of these architectures that are able to demonstrate near expert level performance, but these are just on normal chromosomes. So what about chromosomes that are abnormal? That's what we care about. So there have only been a couple studies that we found that actually look at these recurrent cytogenetic abnormalities and being able to classify those. And those only dealt with a single abnormality, namely the translocation of chromosome 9 and 22, which is correlated with chronic myeloid leukemia and the BCR-ABL gene fusion. And so these studies indicated that CNNs were able to robustly identify the single abnormality, but what about others? And what about in the context of actually automating this in the clinic? Because many of these cases don't contain just a single abnormality, they contain many, and they also contain abnormalities of which they're not recurrent, they're not known. So we decided to add in a couple of additional types of chromosomes rather than just the normal and not just a single recurrent abnormality, but multiple, including deletions, inversions, isochromosomes, translocations, and non-recurrent abnormalities, some in which the derivative chromosome is known. So here I'm showing that there's some sort of unknown abnormality, maybe in the P arm, but it's obvious from the banding pattern that maybe the derivative is known, and then there's some cases where the derivative is unknown. So having all these types, we felt was important for actually benchmarking where an automated classifier might come into play. And so in a data set that assembled many recurrent abnormalities found in AML, and a few that are also found in other blood cancers, we put together what we felt was a pretty good representative data set for a proof of principle study to be able to actually test this. And so there's some nomenclature that you might want to, for the results interpretation, just familiarize yourself. So del, deletion, N is inversion, I isochromosome, T translocation, and dir is derivative. So, and then I'm calling the known derivative unknown D for derivative, and sometimes these abnormal chromosomes are referred to as marker chromosomes, the ones that have the unknown derivative. And so the process flow for this study, at least, was to take 550 karyograms and actually isolate the individual chromosome images out of it, and then split that up into training, validation, and test set. There was some augmentation performed on the training images, including left-right flips, enhancement of image brightness and dimming, and then to evaluate the trained models on the test set. And so the architecture and the training was, if you're familiar with deep learning and how to train models, was pretty vanilla. That we didn't have a lot of secret sauce there. We used a standard ResNet 34 model and atom optimization. We noticed that slightly larger images were needed. A lot of these models will typically scale down the images to maybe say 128 by 128 pixels. We felt that the larger input made a difference. And then these cyclical learning rates, which are a little less known and used in practice, we felt we actually showed it had a small but significant impact on improving performance, about 1.5%. And that's just where you identify some sort of step size, and that's associated with the number of epochs that you would train, and then you actually vary the learning rate between some sort of base learning rate and a maximum learning rate. And that's able to not only help the model train quicker, but converge to a better local minima in the loss function. So overall, the model performed quite well. It was on the test set, it was getting anywhere between 93 and 94% accuracy, and an F1 score, which is a harmonic mean of precision and recall. It's getting between 92 and 94% there, and so we can dig in a little bit deeper on what it wasn't getting. So in regards to the recurrent abnormalities, overall, if you look at the mean F1 scores, most of them are going to be in the 90 percentile or higher. Again, each of these are about 6 images per class, so not the largest test set that we would want to evaluate this on, but encouraging nonetheless. The two that the model performed worse on were the deletion of the Q31, Q35 arm of chromosome 5 and the isochromosome of chromosome 17. And so examining the images in the test class revealed that these had a lot of examples where the chromosomes were highly bent, or maybe did not look like what the representative image that might be found in the training set would be. And so this gave us some confidence that indeed that's a caveat for the model, is training on big chromosomes or testing on big chromosomes could be an issue that could trip this up and need to be ironed out going forward. Another way of interpreting the way the model is doing and learning these ways of making predictions is to use what's called gradient class activation maps, whereby you take the gradients and then give an input image and see which pixels light up, cause the most activation. And those are generally considered to be more important for the model's decision. And so we found, for example, between chromosome 16 and the inverted version, a very interesting feature in that one of the ways the cytogeneticist will identify by eye inversion 16 is by the centromere pointing upwards, indicating that it's been inverted. And so we found in most of our cases for both chromosome 16 and the inverted form that the centromere region was highly activated in all of those. And then for the deletion of 5, we found that both in the normal chromosome 5 and the deletion that the Q arm was highly activated as well. And so while these don't prove that it's learning these as the general rule, it does suggest that it's focusing on areas that are more biologically relevant and not just learning some sort of corollary pattern. And so what about the unknown abnormalities, the ones in which the abnormality is not recurrent? So what I didn't mention in great detail was that for that class, a lot of these abnormalities had lots of different derivative chromosomes. And so, in other words, none of them really looked like one another. And we felt this was a really key component of this because you can train a model on, as I mentioned, there's like 22 recurrent abnormalities for AML. There's many more for all sorts of hematological diseases. And so, in theory, even if you train for all of those, you're going to have abnormalities that aren't recurrent, and you need to be able to have the model do something with it. You don't want it flagging that as a recurrent abnormality and wasting precious time to go review. So having this non-recurrent class was important. And what was interesting was that when you look at the class activation map of this, it's not focusing really on the chromosome at all. So it's kind of, the model's learned to identify that this class, what it actually means is that it doesn't really look like anything else that's in the data set. And so we felt that was an important component for actually trying to, say, commercialize something like this. And when you stack our model's performance up to some of the established ones, it performs as well or better on the normal chromosomes. The only one that it did not perform as well on was one with 87,000 images, although this performance was on the validation set. It wasn't an external holdout test set. And then we demonstrate robust performance on abnormal chromosomes as well. So some future directions of this is that it might be nice to actually use context. So rather than just individual images, a lot of these recurring abnormalities will, as we heard in our last talk, they'll occur in patterns where they accumulate together, or when you find one kind of abnormality, you might find the other. So having that contextual information through some sort of transformer could be useful. And then expanding the number of recurrent abnormalities to be a little bit more comprehensive would be nice. and then integrate some sort of segmentation algorithm. So there are a lot of algorithms that will segment chromosomes, and just putting this into a pipeline that a cytogeneticist could use is something we envision doing as well. And so to summarize, we looked at recurrent abnormalities in AML and APML, and we used a CNN to get accurate classification of these, and showed that classifying with these CNNs they can learn biologically meaningful representations. And we believe that something like this could be used to flag APML, the APML T1517, and expand the scope of how cytogeneticists can actually study these recurrent abnormalities in the clinic. And so with that, I'd like to thank the members of the Kin Lab at UT Southwestern, specifically Daewon and Chanhee Park, for their mentorship and help on this project, and also collaborators in the hematopathology department, Prasad Kaduru and Rolando Garcia, and my funding through Cancer Prevention and Research Institute of Texas. So thank you, and I'll take any questions.
Okay. I'll start with the first question. I think this is super cool. And it clearly has good, important clinical applications. So you were saying that some of these, the ones that the model wasn't able to pick up are like these kind of weird shapes or like slightly abnormal. Do you foresee any way to incorporate like some, perhaps human intervention or something where you can then update the model? Or, I know that it's hard to get enough training data with ones that are kind of in an odd shape, but any way you can kind of systematically improve the model to address these kind of cases?
Right. So there's methods that could be used to say, like if you could identify bent chromosomes, like training a model to classify a bent chromosome versus a straight one wouldn't be that hard. And there's actually correction mechanisms that can straighten these beforehand, or it could just flag it and say it's bent, and maybe consider that before you go spend your time and check it out.
Okay, yeah, no, I think it makes sense. So either pre-processing or maybe creating bent chromosomes to train on or something like this, right?
Right.
Yeah. Oh, here.
Thank you. Great talk. So have you noticed any class imbalance problems from during your training because these genetic abnormalities in AML can occur at different frequencies?
Yeah, it's a great question. Yes, we did have a class imbalance. So the only abnormality that had kind of an overrepresentation was the T922. Everything else we used just data augmentation to generate to increase the number of examples in the training set up to a proportional amount to, say, the normal chromosomes. So they're all about relatively represented in the training set.
Okay. Well, if there aren't any other questions, I think we're kind of right on time, so we could thank Andrew again. It's a great talk.